Hi, let's begin with a fresh system. The next system is excretory system. Clinically important one. Of course, clinically all the systems are important. But why I call this important is also from academic point of view. You get a lot of MCQs from the kidney and renal functions and bladder functions, acid-base balance. So we will go through those topics in the conceptual details. In the kidney, we will discuss introduction, nephron, JJ apparatus, renal circulation, then the clearance concept. Lots of questions have come in the past related to the concept of clearance. Then, of course, GFR, a big topic, factors determining influencing the GFR, and tubular functions, how the substances are reabsorbed, how sodium and water are handled by various parts of the nephron, starting with the kidney. And this is an introductory part. So basic structural and functional unit is the nephron, and therefore, although it will be too basic to discuss the parts of the nephron, but still, just as a quick revision, look, renal artery breaks up into arterioles, and then there is afferent arteriole, which brings the blood into the glomerulus. Glomerulus is a tuft of capillaries. Here, there will be filtration of substances into the Bowman's capsule, and then they will start flowing down the tubules. So we have a proximal convoluted tubule, which is proximal to the glomerulus, and the proximal straight tubule after that. Then we have the loop of Henle, the descending limb and the ascending limb. The ascending limb has a thin portion first, followed by the thick ascending limb, thick wall. So that will be called as thick ascending limb of loop of Henle, which will then lead to the distal part of the nephron. Now the distal nephron starts. So distal convoluted tubule, and then a connecting tubule, which will then finally lead to the into the collecting duct. Final urine formation will happen in the collecting duct. Final pH of the urine, final osmolarity of the urine, all of that will be decided in the collecting duct. And then the ducts from various nephrons, they join to form a duct of Bellini, and then finally these ducts, now the urine has been formed, so it will be drained up to the pelvic callaceal system, and from there, of course, it will be drained into the down the ureter to enter the bladder. So that's how it happens. Let me also add one point here, although we will discuss it in detail later on. But you know, a very interesting type of example for the functioning of the kidneys. Interesting one. How there are these three steps in the urine formation that can be seen. Glomerular filtration, then tubular reabsorption and tubular secretion. Three steps in the urine formation. You know, the example is something like this. Suppose we have to set up a cupboard. There are a few things already there in the cupboard, the books and the other stuff, and we want to clean it up. So what do? We will first take out everything from the cupboard, everything, and then whatever is required will be put back, whatever is not required will be discarded. If you want to add something, that will be also added, and then cupboard will be set up again completely. I had read that example somewhere. Somewhat similar is happening here, that in the glomerular filtration, everything is filtered out except, of course, the proteins in the plasma and the cells. So these two things are not filtered, but otherwise everything is filtered out. And then whatever is required by the body will be reabsorbed. It will be taken back into the blood. Whatever was not required will be secreted by the tubule. Look, glomerular filtration cannot remove the substance entirely. So, we have secretion. Substances will be secreted into the tubules. And finally then, all of this will go on happening in different parts of the nephron. Final processing will happen in the collecting duct and the urine will be formed. So, if I were to say what is the urine composition, I mean it is made up of what? Or what are the processes? So, the processes or the concentration of substances or the final urine composition is equal to glomerular filtration plus tubular secretion minus the tubular reabsorption, isn't it? The filtration and the secretion, these two processes are basically to eliminate the substances into the urine. So, it will be, it is taken into one bracket, minus the substances that were reabsorbed. I mean, that's not coming into the urine, that is taken back into the blood. This is how urine formation takes place by these three steps or processes. And that was the most basic part. Now, let's see the types of nephron. Each kidney is made up of about 1 million nephrons. So, let's see the types of nephron. Now, if you look at the structure of the kidney, it has two parts. There is the renal cortex, the outer part, and renal medulla, that is the inner part. And therefore, there are two types of nephrons. First type is the cortical nephrons. Cortical nephrons, and please take it as a representative diagram. So, cortical nephrons are something like this. They are glomeruli and then the loops. So, 85% nephrons are cortical nephrons. So, majority of the population. They have short loops. They have short loops, you can see there. And what is their function? They will form the normotonic urine. I mean, to concentrate or dilute the urine is not the function of these nephrons. They will only excrete the substances in a normotonic urine. The second type of nephron, which is more of course everything is important, but otherwise functioning wise you should look at the other type, which is juxtamedullary nephron. The juxtamedullary nephron, about 15% population is that of juxtamedullary nephrons. Their glomeruli are located at the junction of cortex and medulla, for which reason it is called as juxtamedullary. It is in juxtaposition with the medulla, as we will see here. So, glomeruli are located at the junction of the cortex and medulla, and they have long loops. That's should be understood. They have long loops which come all the way into the inner medulla. So, let's write it down. Juxtamedullary nephron has long loop, and the most important part is that the concentrated urine is formed by these 15% nephrons. Hypertonic urine, formation of a hypertonic urine, is the function of these 15% juxtamedullary nephrons. right? So these two populations of nephrons can be seen in the kidney. But if you have noted, all the glomeruli are essentially located in the cortex only, right? Let's just add one more point in this about the renal blood flow. Renal blood flow. You know, if you compare the cortex and medulla, this is a comparison of renal cortex and renal medulla. Most of the blood flow goes to the cortex. Maximum, majority fraction goes to the cortex, which is quite easy to understand because all the glomeruli are situated in the cortex. So logically speaking, major chunk of blood flow, if you see the fraction wise, greatest fraction is in the cortex and small fraction in the medulla, the blood flow comparison. But oxygen consumption, if you were to compare the oxygen consumption of cortex and medulla, what do you find? Oxygen consumption is slightly greater in the medulla as compared to the cortex. Not a big difference, but some difference is there, and medulla or consumption of oxygen in the medulla is greater as compared to cortex. The question is, why is that? Okay, note this point that the oxygen consumption in the kidney, or let's say ATP breakdown or metabolic rate in the kidney, is directly linked to or directly proportional to the tubular load for sodium reabsorption. So that's one point that should be clearly understood. The metabolic rate in the kidney is directly linked to the tubular load for sodium reabsorption. And let's talk about a part of the nephron, that is JG apparatus. JG apparatus. You know, we are slowly moving towards the next pattern. Previously there was AIPGE, All India PG entrance exam, then there was NEET. Now we are moving towards the next pattern, and in the next pattern, majority of the questions are likely to be problem solving type or clinical case type. But previously there used to be lot of direct questions, one-liner type. One such question was, which of the following is the name of this cell? Alternate name. Well, first of all, how is it pronounced? I'll try my best. It's called as Gumati cell. Gumati cell. So, where is this cell, and what is this cell? That's the question. And the options are the macula densa cell, the Lassis cell, the granular JG cell, or type 1 cortical interstitial cell. Is known by this name as well, because this is the name of the discoverer who discovered this JG apparatus and that particular cell. Therefore, just remember it. Okay, so JG apparatus. There is one more question, and this amazing question, because I remember it was asked in 2012 or something. The question was, the JG apparatus lies in close relation to, okay, and the options given were afferent arteriole, early DCT, collecting duct, or glomerulus. And I tell you, More than 80% students gave wrong answer to this question. Most of them gave the answer as early DCT, but that's not correct. The answer is actually, hold your thoughts, answer is coming up in 2 minutes. What is JG apparatus? Look, for every nephron, here is the structure of the nephron. For each nephron, what happens is, its afferent arteriole and early part of DCT, early DCT, a thick ascending thick ascending limb of loop of Henle, I beg your pardon, and the early DCT. These two parts, they come close near the glomerulus. So they will come close to each other near the glomerulus, forming a complex of cells which is called as JG apparatus. Okay? And so there are 3 cells in this complex. First, the cell in the wall of the afferent arteriole, the cell in the wall of the afferent arteriole. Second, the cell in the early DCT, so that cell which is lining the early part of DCT, and the third cell is between these two cells, between these two parts, the afferent arteriole and the tubule, the early distal tubule. The third cell between these two. So these 3 cells, okay, afferent arteriole, early DCT, and the cell between these two. And therefore this makes a complex. What is the advantage of this kind of a complex is that there is an easy signaling that is possible from the early DCT up to the afferent arteriole due to the presence of such kind of a complex. Because now the distal part of nephron is beginning. When early DCT comes, now the distal part of the nephron is starting, and that means now finally the urine is going to be formed. So there is a likelihood that suppose lot of sodium has reached this part, then the sodium might be lost into the urine, and that's not what we desire. That's not what is desirable, I mean to say, and therefore it will send the signal to the afferent arteriole and make the required changes, as we'll say, and therefore this mechanism is made easy by such kind of a complex of cells. Okay, so that's the significance of the JG apparatus. Let's see the cells in the JG apparatus. First, the granular, or also called as JG cell, which is in the afferent arteriole. Granular or JG cell. What is its function? It synthesizes the enzyme renin, actually prorenin, which then forms the renin. So, what is the importance of this? This is involved in regulation of blood volume and blood pressure. We have done this in the CVS, so I'll just avoid the repetition. That if there is a decrease in blood volume and therefore there is a decrease in blood pressure, then there are volume sensors and baroreceptors present in the renal artery. They will sense it, and then these cells will release renin. There will be something called as PRA, plasma renin activity. It will increase, and what happens as a result? It will convert angiotensinogen into angiotensin 1. And then while it is passing through the lungs, there will be angiotensin 2, and also it will cause, angiotensin 2 will cause release of aldosterone. So that is the significance of RAAS, renin angiotensin aldosterone system. So that's the first function of the component of the JG apparatus, regulation of blood volume and blood pressure. I would urge you to refer to the chapter on CVS, regulation of blood pressure, so that you can correlate this renin, angiotensin, aldosterone system. Second cell is the cell lining the early DCT. Many authors believe that it is in the later part of the thick ascending limb. So, thick ascending limb is ending and the early part of DCT is starting. The cell is located there. And it is called as the macula densa. Now, what is the function of this cell? This is the major component, and the main function of the JG apparatus is performed by this cell. By the way, the question that I asked you, the JG apparatus lies in close relation to, and the options given are the afferent arteriole, early DCT, collecting duct and glomerulus. These four were the options, and majority of the students gave the answer early DCT because thinking that macula densa is the major component, so maybe. No, the answer is glomerulus because the name is juxtaglomerular apparatus. Why is that? That's because it is in juxtaposition to the glomerulus. Many students forgot this and they gave the wrong answer. So, let's come back to this. Macula densa, the cell lining the early DCT. What is the function of macula densa? Sensing of sodium and also chloride actually in the tubular fluids reaching the early DCT. Sensing of sodium and chloride in the tubular fluids that are reaching the early DCT, and then what it does is to initiate the tubuloglomerular feedback. Tubuloglomerular feedback, very important mechanism for the body physiology, very important mechanism. Why do I call it important? See here. Let me explain this mechanism in detail. You know, we, the human beings, are facing lots of situations on a daily basis and on a moment to moment, minute to minute basis, whereby the blood pressure keeps on changing. I mean, change of posture, exercise, stress. The blood pressure keeps on changing so frequently. Therefore, the renal perfusion pressure will also keep changing. It can change. Of course, there is auto-regulation of blood flow in the kidney. That should not be forgotten. Yet, there is a possibility that if there is increased blood pressure, systemic blood pressure, and therefore there is increased renal blood flow by any such reason, that the blood pressure has got altered and therefore there is increased renal blood flow. Well, increased renal blood flow will mean increased GFR. I mean, that should be obvious, and we will discuss about it later when we discuss the glomerular filtration rate. But more the blood coming into the kidneys, more the blood coming into the glomerulus, more will be the filtration at the glomerulus. So, GFR increases, and if the GFR increases, there will be increased filtration of sodium. And if there is increased filtration of sodium, it will be reabsorbed from the PCT. But imagine that there is increased flow of tubular fluids and lot of sodium has got filtered and it has come to the distal nephron. Now there is a real possibility that this sodium might be lost into the urine because it could not be adequately reabsorbed. Sodium will be lost means water also will be lost. So, we are looking at a possible fluid and electrolyte disturbance. Yes, there is a possibility of sodium loss, water loss, and resulting in fluid and electrolyte abnormality or disturbance. Can you imagine this? I mean, why did I say it's a very important feedback mechanism? Is because just by change of posture and change of blood pressure, if this were to happen, we could not survive. It will be a complete disaster for the body. But thankfully, we have a mechanism which is in the form of tubuloglomerular feedback. That means from the tubule to the glomerulus, a signal is being sent, and this disturbance will be prevented. That's a tubuloglomerular feedback. How is it prevented? Look, as I mentioned just now, sodium is reaching the early DCT. Okay. Now, these macula densa cells, they are going to be responsible for tubuloglomerular feedback. How do they achieve this? Let's say here is a macula densa cell in the early DCT. Now, sodium has reached in excess amounts in this part of the nephron, and I already mentioned that sodium reabsorption occurs by two steps. This sodium has to be first from the tubule, from the lumen, it has to enter the cell lining the tubule. There are transporters NKCC or NCC, and then on the basolateral membrane there is sodium potassium pump, right? Sodium potassium pump, which will then take the sodium from inside the cell, and finally sodium gets reabsorbed into the blood. Now we are looking at that tubuloglomerular feedback. How will it be initiated? So, when there is increased tubular sodium, it will be sensed by macula densa cell. As we have already mentioned, that's the function of macula densa, sensing of sodium. And then sodium reabsorption, as it goes on happening, increasing amounts of sodium reabsorption. That means increased activity of sodium potassium pump, right? Increased activity of sodium potassium ATPase means what? Increased breakdown of ATP. So, increased sodium reabsorption meaning increased ATP breakdown by virtue of activity of the sodium potassium ATPase. Increased ATP breakdown means what remains? ADP, AMP, and finally adenosine. So, there is increased formation of adenosine. And this adenosine is the important ligand in the tubuloglomerular feedback. So, tomorrow they can ask you, what is the ligand involved in tubuloglomerular feedback? One ligand is adenosine. Now, this adenosine which is formed here during the reabsorption of sodium and the ATP breakdown, this adenosine will go to the afferent arteriole and it will cause constriction of the afferent arteriole. Adenosine will cause constriction of the afferent arteriole means blood flow to the glomerulus will decrease, and therefore GFR and sodium filtration will also decrease. That was the demand. because sodium was otherwise it was going to be lost into the urine. So adenosine is one important ligand for this. I want you to understand one point. Adenosine is a vasodilator everywhere in the body. But in the kidney, in the renal circulation, it acts as a vasoconstrictor. Please make a note of this. Adenosine is a vasodilator, and it's an important vasodilator everywhere in the body. Let me just quickly tell you that, for instance, suppose myocardial metabolic rate has increased and it needs more blood flow in the coronaries. What happens is there is quick ATP breakdown because metabolic rate has increased. ATP will be converted to ADP, then AMP, finally adenosine is formed. That adenosine leaks out of the cells and it will dilate the blood vessels, coronary vessels. So blood flow will increase into that part. This is based on the Bernays hypothesis. So that's how blood flow can increase to the part. But in the kidney, it leads to vasoconstriction. And that's obviously because we have two types of receptors, A1 and A2. So generally the A2 receptors elsewhere, and in the kidney there is other receptor. So adenosine is the one ligand. The other one is ATP. The other signal is generated by the ATP itself. We know that the ATP is a neurotransmitter. It acts like a neurotransmitter or a ligand. So here it's acting as a ligand, and ATP acting via its receptors called as P2X. These are purinergic receptors. These are called as purinergic receptors. So purinergic receptor of the adenosine triphosphate, and therefore even ATP can initiate this tubuloglomerular feedback, and the effect is constriction of the afferent arteriole. So less blood will come into the glomerulus, and therefore less glomerular filtration rate and less filtration of sodium. So sodium will come less and less into the tubules. That is how loss of sodium and fluid and electrolyte imbalance will be prevented. That is the function of tubuloglomerular feedback. Since it's outside the blood vessel, it's called as and outside the glomerulus. So extraglomerular cell or mesangial cell. It's called mesangial cell. This is also called as Lassie cell. The Lassie cell, or I have mentioned this name already, GOORMAGH cell, Goumati cell. Yes, we very often we tend to pronounce each letter in the word, but that's not how most of the words are pronounced. Okay. Now this, what is the function of this cell? This also is involved in the TG feedback. It can alter the vessel diameter of the afferent arteriole, and this is also involved in the immune mechanisms related to the JG apparatus. You will learn this in pathology about the MPGN, RPGN, glomerular nephritis, where immune complex formation, immune complex deposition, this cell is involved. Anyways, but that's the Lassie cell.